Hello and welcome to day 7 of our class on RNA sequencing. So this is video 3, differential expression using DE-seq. So a lot of you have worked with some data so far this week, and hopefully you've looked through Aaron's videos and learned a little bit about differential expression. I want to talk about the fact that fold change is not a very good metric when you're looking for genes that have changed significantly. This is not something I understood when I first started doing short read sequencing or microarrays. I assumed that if something had fold changed more than 2, that was significant, and I'm going to show you why that's not true. So if you take a look at this plot over here, you'll see that what we've plotted is fold change on this axis and expression on this axis. Every single one of these dots is a gene. And so what you're looking at is how much fold change you have versus the expression. So if you look at this plot, I want you to think about it for a minute and say, now what would you decide had been differentially expressed if you had used a fold change of 2? So this is in logs, so fold change of 2 is going to be about here. And what you see is if you draw a line about there, you see a lot of very low expressed things being fold changed more than 2. That's something known as sampling error. So when you have, say, a room full of balls and you have lots of different colors and you only have a few, maybe 10 that are blue, and I tell you to go pick 100 out of the room, each time you pick it you have some probability of picking a blue ball. But because it is very rare, each time you go to pick it, there will be large differences between it. So one time you may get 0 blue balls, and one time you may get 2 blue balls. And therefore, that's a very big difference in the number of times you've seen the blue balls. This is also true for expression data. So anything on the very low end of expression, you're going to naturally expect a twofold change. What I didn't tell you about this graph is this is 2 replicates graphed against each other. So even when you graph 2 biological replicates, you're going to see some things changing at least 2-fold. And that's, again, because of sampling error. It's going to be very low expressed things in general. You'll also notice that over at this end, at the very high expressed things, they don't fold change very much. So if you have 10 million copies of something in your sequencing data, you don't often expect that in the treated sample or the mutant that you see 20,000 copies. And that's because the higher expressed it is, the less likely you are to get that high of a difference. And yet some things over the high expression are likely to be differentially expressed, but they aren't expected to be 2-fold differentially expressed. They can still be differentially expressed. So this is what it looks like when you graph real data. This is my data, that is the difference between non-drug treated and drug treated gross-seq samples. And what you see here is you have a lot of red dots, and the red dots are what DE-seq is calling as differentially expressed. And you notice that over on the low expression end, it does not call many things. Again, that's because it's hard to tell random noise from real differential expression when you're low expressed. Sort of like if you were trying to do a Western blot on a protein that was so low expressed you could barely see that band. Each time you did it, you might have a little bit of variability, and so it could look like things are changing quite a bit between replicates. So the fact that fold change is not a good metric is what led a lot of people to come up with many different programs to estimate the dispersion of biological replicates, basically. So what they're trying to do is, if this is the graph of 2 replicates against each other, you expect some noise down here, but you of course want to know what your real differential expression looks like, which will be up here or down here. So all of these programs over here are attempting to draw basically a line where your natural variability is, so that if there's anything outside of natural variability, that's what's called as differentially expressed. I've only used DE-seq and BASE-seq from this list, but I suggest that some of the other ones may be more useful to you. You should look into them and decide based on your experiment which one would be most proper. The only reason I used DE-seq was at the time I started doing my experiment, only EdgeR and DE-seq were available, but now the options that you have to you are far more. So again, they're all trying to just draw this line, and they're using different mathematics in order to attempt to draw a line where natural variability is, so that you can call things outside of natural variability. So I'm going to walk you through one DESeq, but I would suggest that if you're going to do it yourself, you actually go back to the manual, because the way I'm doing this was the original way that this DESeq suggested it be done 2 years ago, and they've made several updates. There's even a DESeq 2 now that I haven't used, but I expect that those programs are far better than the originals, since they've had some time to optimize. So go to the manual. That's a great place to learn it. There's also a blog that I thought was pretty good that walked people through both DESeq and DESeq 2. Remember the assumptions of each of these differential expression programs, which is the assumption for DESeq is that most genes are not changing. That's a very important assumption, because if, say, every gene in your genome has been upregulated, DESeq will not see that, and I'll show you that later. Also, DESeq uses R. I think that once you've learned how to use Unix, R is a very good next step, because R is a strong statistical package that basically, once you've processed your data into a number per gene or a number per seq site, R can be used to do a lot of very cool stuff. It's got great graphing ability, and it's fairly easy to learn. So check out R, go to the rproject.org and get some information. The only challenge with R is because the program name is R, it's really hard to Google for command line stuff. So if you're going to try and look up how to do something in R, I'd suggest going to the website. Okay, so DESeq and many of these other programs start with this file. Basically, it has a list of genes or exons or any region that has a count of reads. It's got all your samples, and then it's got a tab delimited file with the number of counts for each sample at that, say, gene. Now, this is the information that DESeq uses. There's a couple of ways to get that information. HTSeq and Pasilla are apparently two options. However, I've built my own program to basically take a BAM file and count the reads using bed tools, and then build a table that can then be inputted into DESeq. So you're going to have to find a way to get to this table before you start. But for the class, I'm going to give you one of these tables. Now, in my table, I want to let you know that I have a lot of information, far more than what DESeq needs. So I've got the number of reads at every gene, I've got FPCAMs at every gene, I've got other statistics for every gene. You don't need that much information, but it will be in the file I give you. If we zoom in, you can see I've basically got a lot of metadata on every gene for every single sample. So I'm just letting you know that because there'll be an extra step because I'm loading in excess data. So the steps in DESeq is that we're going to read in the data, we're going to label the data, we're going to normalize the data for the number of reads in each sample, and I'll show you why that's important in a minute. We are going to estimate the variance, and I'm not going to go through other quality control steps, but you really should when you do yours. Just for brevity, I'm going to go through the main steps. And then it will use a negative binomial, which is the mathematical way of looking for that line I was talking about earlier. So to load R, at least while we're in class, we're going to use this command line, which is basically just using QSub to get us onto one of the children nodes and pipe the visualization through. Remember that we are setting wall time to 3 hours, so if you're taking longer than 3 hours, your program will just die on you. And we're using the sread queue, which is only available during class, as is this -W flag. That's only available during class. So if you're doing this after class, don't use the -W and the rest of this line, and don't use the sqread. So we've typed in this line, it then uses QSub to get us onto one of the children nodes. I'm on node 17 right now. I'll then start R up by typing in this command. You can, if you know R a little bit, you know you can just start R by typing R, but that will take you to the whichever main R is installed. And on Vieques, this is the only R that has the DESeq library installed. So that's why we're using this R. So type out this command and hit enter, and that will load R. In case you're curious, the reason that it is named R is that there was a statistical package named S that was very powerful. This was the package created after it, so they decided to just keep going down in the alphabet and naming it R this time. So you type R and it loads R, gives a bunch of information. You then have to load the DESeq library. Again, it's only installed on this R, so make sure you're using the correct R version or it won't load. So by typing library DESeq, you've now loaded R, and it gives you a bunch of information about the load. It will give you a couple of errors that are not a big deal. They're just a couple things that got reinstalled separate times. Okay, so then after you've loaded the DESeq library, you need to load in your data. You do this by typing data, a little caret symbol, and a line, which makes an arrow. Read delim, and then the file name, and then you'll need to give it a little more information about the file you're reading in, which is that it is tab separated. That's how you indicate this, and that there is a header in the file I'm giving you. Again, the file I showed you earlier is the one that you're loading in here, and the one we will load in class. Now remember that that has a lot of metadata on a lot of different samples, and yet I only want to compare 2 of them, and therefore the extra step that I'm adding in here is that I'm going to build a counts table that only selects 2 of those columns. So I'm telling it to choose column 8 and column 10 from this data, and that will be my counts table. And then if I look at the counts table by typing it, it will say that these lines have these numbers for that sample and these numbers for that sample. Now you'll notice that I don't have a gene name at this point. That's an issue. We need a gene name. Now I had a column in this file data called name that had the gene names, and therefore I am going to tell counts table that the row names are in that data file under the column that says its name is name. So I am calling the name column and then putting it in as row names. If you were again to type counts table, it will now give you the gene names where those numbers are. I also need to tell it what conditions I'm using. So in this, I'm using a combine function in R, and basically I'm just telling it I have 2 samples, a DMSO and a Nutlin. So now it knows the conditions I'm using. And then we have to load all of this data into a CDS. This is a it's sort of like a setup vector system that knows what type of data you're going to need to give it. And therefore, you can start telling it things it needs to know. And it really needs to know 4 different things. It needs to know what your count table is and what your conditions are. It also needs to know what your estimated size factors are. So estimated size factors, let's say you sequence 2 samples and one of them has 4 times as many reads as the other one. That's an issue when calling differential expression, and you need to correct for that. Now you can correct for that by telling DESeq how many more reads were in one sample than the other, or you can just have DESeq take your counts table and guess at that. So here I've had it guess at how many counts, or how much more sequencing there was on the one sample than the other sample. And then after I've had it estimate the size factors, I ask it for what did it estimate, and it spits out that for this sample it thinks that I'm going to call it around 1, and for this sample I'm going to call it around 0.95, meaning that they were sequenced to a fairly equivalent depth. Now always check these size factors. I'll show you why later, but if you know that you have 4 times as many reads and this number comes out anything but 4 times as many reads, that's a problem. And you can force DESeq to use the numbers you give it for the size factors. So what does DESeq do with these size factors? Basically, it makes a list. The way it calculates them is it rank orders all the numbers that you gave it in that file, finds the median, and then sees if the median of one sample uses the median of one sample and the median of the other sample to tell you about how different they are in read depth. At least that's the original way DESeq did it. They may have updated it at this point. So after we've had it sort of use these size factors to normalize how many reads are in both samples, we need to estimate the amount of dispersion, which is the line below. Oh, I should tell you, you can type CDS after every time you input something into CDS, you'll get the information that it currently has. So I've typed CDS, and right now it has featured data. I'm going to estimate dispersions and tell it which method to use, which I wouldn't suggest these ones unless you have no replicates. I happen to be running this on a sample that I have no replicates for. But go look at the different methods under estimated dispersions and choose the correct method for what you have. So there'll be a correct method for if you have replicates or not, and how you want it to estimate the dispersions. But then after you have estimated dispersions, if you type CDS again, you'll see that where feature data was none before you estimated dispersions, now feature data has a lot of different information inside of it. So CDS just sort of gives the computer the information it needs in order to do the differential expression calculation it's going to do. So we have estimated the size factors and we have estimated the dispersions. Those are the four things it needs. The counts table, the conditions, the estimated size factors, and the estimated dispersions. And then it is ready to calculate results for you. So to calculate results, I've named my results res, and I'm going to use a negative binomial test in order to get me results. I'm going to give it all of the information it needs by feeding it CDS and telling it the two samples I want to compare, DiaMeSode and Nutland. The reason I have to do this is when I gave it conditions, I only gave it 2, but you can put in 10 different conditions and then tell it to test just these 2. You know, if you have 10 different ways you've treated the samples, you then tell it to test the 2 different ways against each other. It can only test the 2 different ways against each other. If you do have 10 different methods of treating the samples, you may want to try Bayes-Seq. I haven't used it, but I've been told that it's able to handle multiple sample type comparisons. So now I've typed in res, and I actually have the results under res. So if I type head into res, I can see what the first 6 lines of those results are. Basically, it's saying for these genes, I have this base mean. So remember when I told you it was normalizing. It will take the number that was, say, 369 before, and then take that estimated size factor and adjust how many reads it is based on how many reads total there were. So you'll get an average base mean, which is the reads across both samples. And then you'll get the base mean A, which is reads against the DiaMeSode, and base mean B, reads against the Nutland in my case. You'll get the fold change, the log 2 fold change, the p-value, and the adjusted p-value. The adjusted p-value is very important in DSeq. So it's going to give you a p-value, but if you're only doing one test, the p-value might look pretty good. But when you do thousands and thousands of chances or tests, you're going to have thousands and thousands of chances of making an error. And therefore, you must adjust the p-value, and you cannot use the p-value. You need to use the adjusted p-value to decide if something is likely to be a legitimate hit. And you'll notice that all 6 of these have a value of 1, which means they are not likely to be differentially expressed. So once you've got the results, you're probably going to want to plot to see how they look. So here's how I built the plot function. I don't think you have to do this anymore. I'm pretty sure DESeq now has a function in it that will draw a plot from your results, but you'll have to look into the manual to find out that. Once you have the plot, you want to save it, and we actually haven't created it at this point. At this point, we've created a function to make a plot, but we have not made a plot yet. You're going to want to save this plot, so you're going to open a device called PNG, which is a type of picture file, and tell it that you want to name it whatever you want to name it, say filename.png, by using this command. This command will then plot using that function that you created a slide ago, but it needs to save it into your file. So since the PNG is considered a device, you tell it turn the device off, and by doing that, it will then save your plot into this file. The other thing you're probably going to want to do is write that data we found before into a file so you can access it later. So to write the results into a file, you're going to do write.table, give it the data, the results, not the data, the results, and then tell it what file name you want to put it into, and you'll probably have to tell it that you want to append false and that the separator you want to use is tabs. So it will create a tab delimited file for you of your results. Oh, the file name, I didn't do it here, but the file name has to have quotes around it. So another thing you can do is tell it to only output the significant genes. So here I've taken my results and said, basically take the results, but only give me the results that have a p-value of less than 0.1. So those ones are likely to be pretty good results, and I'm going to shove them into a new vector named resSig. So results significant has only things that have a p-value of less than, have an adjusted p-value of less than 0.1. So here I have written all of my results, so you can see that because I've got it writing all of my results to this file named append is equal to false, and separation is equal to a tab delimited separated file. I tend to write all of the results into one file and then all the significant results into a second file so that I can just look at the significant genes if I want to. So that's got a different file name, but now it's going to write the significant results. And then I tend to write a third file too. I don't use this one as often, but it's kind of nice if you need to flip through real quick, which the third file is the significant results we're writing, but we're going to order them by the p-value so that the things with the smallest p-value end up at the very top of the list, so we can find them very quickly. And it's the same format as before. Give it the file name, append is equal to false, and the separator is tab delimited. And then you'll have a file with your results in it. Okay, so a couple of notes. Make sure that you run all the quality control options that are in the manual. I didn't go over them here just for brevity's sake, but it's really important to make sure you have quality data, or DESeq may not give you, it'll give you results, it just may not be the right results. The other programs I listed at the beginning may be just as useful. I only use DESeq because that was what I used when my data was available 2 years ago. I'm going to get some new data soon, and I'll probably be looking into several of those to see which one I think is most useful for my data. I need to make a note that a p-value of 0.0005 does not mean that the gene has changed in expression. A lot of people are confused about what a p-value means, and they assume that if it's below their cutoff, then it must be a real change. Remember that a p-value is just the chance that that gene could have the expression change that we saw, and still the two samples were not different to each other. So the way I think about it, it's not exactly correct, but the way I think about it is that's the percentage chance that if I put two replicates in, it would still have that large of an expression change. Make sure that if the plot looks funny, not like the example we showed, that you try and understand why. Because probably if your MA plot looks funny, it means there's something wrong with your data, and DESeq is not going to give you the right answer if there's something wrong with your data. The last thing I want to say is to remember your assumptions. So I'm going to give you a way that I've seen that DESeq will still give you a result and be wrong at the same time. So if I were to take only these genes that are actually differentially expressed, go back to the original file and filter the original file to throw in only genes that were differentially expressed, which, I'll tell you why in a minute. That would be something you would do. If you then run it through DESeq, you'll see that these red dots aren't red anymore. Most of them go back into the black. The reason for that, remember, is that we're normalizing. So if you give it nothing but genes that are differentially expressed, it will normalize the two lists because it wants the same number of reads in the two lists. And you will end up with none of them being called as differentially expressed. The place I ran into this is I was working with a group of RNAs called eRNAs, and in my sample, all of the eRNAs expressions go up. And so this is the actual plot for when I fed it to DESeq. Even though all of them are upregulated, because it does that normalization where it tries to match the read numbers, it makes it look like most of them aren't normal or aren't the same. So I would suggest make sure your size factors are what you expect them to be. So in this case, I was expecting size factors of approximately the same, so 0.95 and 1 like we saw earlier. But the size factors when I actually looked at it were 0.7 and 1.4. So I knew that something was wrong, and that thing that was wrong happened to be that all of the RNAs were going up in expression. And that breaks DE-seq's rule, because remember, DE-seq's assumption is that most genes are not changing. And if you break that rule, you will not get the right results. All of these dots should have been red, and yet hardly any of them are. One more thing to note, once you get all of the commands working in your R script, you can actually build, or in your R program, so typing them in manually, you can build an R script, which is that you take every command you've typed and put it into a file named .r, any name .r. Your first line of this file should be sync with the file name that you want to output anything that it would have put on the screen. And then the last line should be sync and just a closed bracket. And what that does is if it was going to write anything on the screen, it will instead write the thing to the screen into this file. Once you have this file named .r or outfile.r, whatever you want to name it, you can run the R script using opt_r this information, so that tells it to run it using this R, and then you're going to tell it, I'm giving you a command line batch script. Don't save any of the information other than what you tell it to save in the script, and don't restore anything from the previous R run. And then this out file name is whatever you named it, name.r. And that will cause R to run it automatically. Why would you want to do that? Let's say you're going to run it on 40 different samples, and all you got to do is change the names in the samples. And then you can build a script that you can just change that name in the script and rerun it several times, and it's much easier than manually inputting it to R every time. So that's about the bare minimum you need to know about DE-seq. If you're going to use it, make sure you read the manual. There's a lot more information in there. I wouldn't take this as a cheat sheet because my data is probably not like your data. So make sure you understand what it's doing and use it in the way built for your data.